Hi guys, welcome back in another video tutorial. Today we will going to talk about some important things that you should remember whenever you are doing primer designing in PCR. Okay, so I have seen various important questions in previous year exam related to that primer designing. So I have decided to make a video on this. After the ending of that video, I will also going to discuss about those questions. Okay. So the first criteria that you should keep in your mind, it is melting temperature. Okay. So what do you mean by the melting temperature? Melting temperature is the temperature at which half of the double stranded DNA is converted into single stranded DNA molecule. Okay, and it indicates the stability of the duplex, and this is the definition of the melting temperature. But this is not a important thing. The important thing is to understand how melting temperature is necessary, and we should keep in our mind about the melting temperature whenever we are designing a primer. So first thing I want to say that why we are designing primer: to anneal them with our templates, right? And there is a particular type of temperature known as annealing temperature that is important to keep in our mind during the primer designing, because at that particular temperature the primer will anneal with the template DNA. Okay, and now this annealing temperature is related with that melting temperature. Okay, how? First of all, the melting temperature will decide at what temperature the primer will anneal to the template and at what temperature the primer will be dissociated. Okay, and that is why it is suggested that the two primers you are using, because we all know that in PCR we are using two primers, one is forward primer, another one is reverse primer, right? And these two primer should have similar melting temperature. Why? Because suppose two primer have, let's say this one has 60° centigrade melting temperature and this one has 40° centigrade melting temperature. That means what? That means at 60° centigrade the primer will be annealed, this primer will be annealed, and at 40° centigrade this primer will be annealed. That simply means that we have to set two different temperature for just dissociation of the two primers, or if we try to say it oppositely, for annealing of the primer, right? And that is the reason why it is often recommended to please keep the melting temperature of these two primer identical. That means, let's say if this primer's melting temperature is 60° centigrade, then it should have like 58° centigrade. That means the temperature difference, the melting temperature difference between the two primers should be less than 5° centigrade. Okay, and if it is occur, then they will anneal near about at the same temperature and they will also dissociate at the same temperature, and amplification can be done just by keeping the temperature at only one value for them. Okay, and that is very important thing, right? So what should be that melting temperature, how we can determine the melting temperature? So first important thing is that I want to told you that melting temperature is highly depends on the GC content. Okay, and there is a formula for determining near about 50 to 20 nucleotide double stranded DNA TM value, that is this one, 4 into G plus C plus 2 into A plus T degree centigrade. Okay, and I will also try to explain you with the help of an example how you can use this formula. So the melting temperature 52 to 58° centigrade is the appropriate one, and we are also using 65° centigrade but above 65 degree centigrade could be detrimental, because remember, above 65 degree centigrade melting temperature will promote secondary annealing. Okay, now I hope you may ask that why 50 to 58? You may also take 40. Why you are not taking? So the idea is, yes, if you going to increase the temperature of the Tm value too much high, it will promote secondary annealing. Similarly, if we use a melting temperature very low, let's say 40-30, then it will promote mismatch hybridization. That means primer can anneal with any of the sequence except the desired one. Okay, and that is not preferable, right? That is why we are taking 52 to 58 degree centigrade as a barrier. Okay, clear? Now let's talk about how you can use this formula. So first of all, let's say this is a sequence A, T, G, C, T, T, G, C, A. Okay, 5 prime to 3 prime. Although here I have not followed the rule of GC content that I will going to explain in the next topic, there I will explain. Okay, so just count the number of GC 1, 2, 3, 4. Okay, that means your value will be 4 into 4. Now count the number of 80 nucleotides 1, 2, 3, 4, 5. 5 into 10, 5 into 2, that means 10, right? Let me remove it. Okay, that means 16 plus 10, your melting temperature for that sequence is 26 degree centigrade. Okay, then yeah, I hope you can understand that why we need to keep GC in our mind, because here we have taken only 4 nucleotide out of 1, 2, 3, 4, 5, 6, 7, 8, 9. That means quite less than half, right, of the total. So that is the idea. That means GC rich sequence will increase the melting temperature. Similarly, the sequence which lack GC will reduce the melting temperature. Okay, so GC content will going to play a very important role. So 40 to 60% of the GC content is preferable. Okay, and according to the equation that I have taken, 4 out of 9 is quite a good term, because it is, if we going to use the percentage, okay, that means quite 44% was my GC content in the sequence that I had taken, right? So that is quite preferable, acceptable, right? So still, as we have taken 44% GC content, it gives us a melting temperature of 26 degree centigrade. And similarly, remember, as we have taken 9 nucleotide only, it also reflects in its melting temperature. That is why length of the primer will also going to play a important value. We will going to discuss about this. So I have told you that 40 to 60% of the GC content is preferable. Another important thing is, suppose let's say this is your primer, and at the 3 prime among the last 5 nucleotides there should be maximum 3C or G. Okay, in some literatures they are also telling that out of 5 there should be 2C or G. Both are correct. So now why we are keeping 3 base, maximum 3 base, but above 3 base is not preferable. Okay, we will going to talk about this. So maximum 3 base out of 5 should be C or G. So if we keep maximum 3 base C or G, then it will promote specific binding. Remember it very very carefully, because we all know that G in between G and C there are 3 hydrogen bond, right? So they will specifically bind with our target DNA. Clear? But similarly, if we use greater than 3G or C, then it will create problem. Okay, because then it can promote mismatch hybridization or another important problematic things that we are not preferring during PCR. Okay, so just remember about that thing, that maximum 3 base at the end or at the direction of the 3' end should be C, and this type of phenomenon is known as GC clamp. Okay, but 2 more GC clamp is not preferable. Out of 5, if you take 4 C or G, then it can create problem. Okay, and another important thing, in the whole primer your GC should be uniformly distributed. Okay, if you keep GC in cluster at a specific position, then they can promote mismatch priming, and that is not preferable. So in that picture we have learnt about the melting temperature, where we have discussed that the two melting temperature of the primer should be similar, so that we can perform the association and dissociation of the primer reaction at the particular temperature. Okay, we do not need to take various temperature for various processes. Okay, 50 to 58 degrees centigrade is preferable. GC content should be 40 to 60%. Okay, 40 to 60% means number of the GC in the total primer in terms of percentage. Okay, that means if your nucleotide is 100, then out of 100 there should be 40 or 60, 40 to 60 GC content will be present. Okay, and another important thing, at the 3' end, please keep at least 2 C or G or maximum 2 C or G. Okay, they will help you for specific binding, but please don't go above 3. Okay, because if you go above 3, then it can create problem. Okay, and your GC content should be equally distributed throughout the primer. Okay, if you use a cluster of GC, then it can promote mismatch priming. Now another important thing, during the numerical that I have done to explain you that formula, I have told you that as we have taken 9 nucleotide, that is why we have obtained the GC temperature of melting temperature 26 degrees centigrade. Okay, so that is why we should take near about 20 to 24 nucleotide, so that we can match this TM value. Okay, and so if you take a 20 to 24 nucleotide primer, and among them if you take only 10 nucleotide G or C, okay, then you can match this melting temperature. Okay, not only that, if you take too small primer, then they can promote mismatch priming, because remember, primer is nothing but ATGC and your DNA is nothing but ATGC. Okay, that is why if you take too small primer, then the specificity will be lost. Okay, because we are constructing the 20 to 24 nucleotide primer having the nucleotides that are specifically complementary to the 3' end of the template. Okay, similar will be in this case. Okay, but if you take only, let's say, 4 nucleotide or 6 nucleotide, then it is possible, right, that ATGC will match with the other part of the template, right, and that will promote mismatch priming. Okay, so that is why the length should be 20 to 24 nucleotide, then it will help you to match the TM value, that is demanded TM value, to get a good primer. Not only that, 20 to 25 4 nucleotide will ensure the specificity of the primer. Okay, now what will happen if we use a too long primer? So in that cases, similar things can happen, that specificity will fall. Because remember, suppose let's say you have made a 40 nucleotide primer. Okay, first of all, it will not help you to match the TM value, the demanded TM value to get a good primer, that is 50 to 58 degrees centigrade. So now let's try to think about that among 40, if you take 20 GC, then 4 into 20, that means 80. There is already 80, right? That means you cannot match the required TM value. First thing is this. And the second thing is, among 40 nucleotide, there may be possible, suppose we are taking a DNA of, let's say, 200 kb. Okay, so there can be possible, right, that among 40 nucleotide, the primer can find complementarity with near about 30 nucleotide and other part of the DNA except the 3' end, right? Because remember, suppose this is a template and this is a wrong template, that means this is a identical this is a similar one with the 3' end of the DNA but not the specific one. Now primer found complementary with that part, so they will anneal, right? Despite these two parts are not complementary, but that will not create any kind of problem. And we have discussed this kind of thing earlier during the site directed mutagenesis, where we have used a wrong primer intentionally, right? But still they will anneal, right? So similar situation can be applicable for these things also. That is why too much long nucleotide primer are not recommended, okay, because it can promote mismatch priming, mismatch hybridization. Number one, it will not help you to match the TM value, okay, that is the thing, okay. And another important thing is, let's say in the 40 nucleotide there can be a chance of repetition, okay. Suppose at 15 number position you have T. After that from 25 you have again A. Okay, and as a result, as they are complementary, there can be a possibility that they can form a secondary structure, and if it happens, then they cannot bind well with your 3' end of the template, and that can affect your PCR yield. Okay, that is why the length of the nucleotide is kept 20 to 24 nucleotide, okay. And among that 20 to 24 nucleotide, it is very important to take less than 4 nucleotide which are repetitive, okay, or which can promote self complementation, self hybridization you can also say, or same cause which can promote self complementarity, okay. So that is the thing of length, okay. So we have to take 50 to 58 degrees centigrade melting temperature, 40 to 60% GC content, and 20 to 24 nucleotide length, okay. And at the 3' end, among 5 nucleotide there should be maximum 3C or G, okay, but not more than that, okay. Now let's move to the next part, that is, yes, primer should not be complementary to themselves or partner primer. That means, suppose you have constructed a primer that is look like that, so what will happen? They can promote a hairpin loop structure formation. Why? Because A can form a complementary reaction, A can form a complementary reaction with T, and this A and T can form complementary reaction with T, and they can form this kind of structure known as hairpin loop, okay. So this is avoidable, right? And another important thing, the primer should not be complementary with its partner primer, because we all know we will going to use 2 primers, one forward primers, another one is reverse primer, so they should not be complementary with each other, okay, especially the 3' end of the primer, okay. That is why I have told you that please do not take more than 3 nucleotide C or G, because they can promote complementarity, and as a result they will pair with each other and leads to the formation of primer dimer, and as a result no primer will bind with your template and you will get nothing after the PCR. Okay, so that is why it is often recommended to please keep extra attention towards whether your primer have the ability to form self complementarity or whether your primers are complementarity with each other. Okay, and I have already told this thing that some nucleotides that are larger than 4 repeats or palindromic regions should be avoided. Okay, palindromic regions means, suppose these things are palindromes. If you try to read this from this direction, the thing you will get, if you try to read this in this direction, the thing you will get will be exactly the same. Okay, that is the meaning of the palindrome, because then they will promote primer dimer. Okay, so that is the some important things that you should keep in your mind during the PCR designing. First of all, melting temperature, that should be 52 to 58 degrees centigrade. Length 20 to 24 nucleotide is preferred. Then please keep maximum 3 G or C at the 3' end. Okay, and primer should not have the ability to form self complementarity, and primer should not be complementarity with each other. Okay, because at first case they can form secondary structure and at the second case they can form primer dimer. Both are not preferable. Okay, and primer should not have such kind of sequence that can promote self complementarity. Okay, now one type of question I have already discussed with you, that is what will happen if we use a long primer. I have discussed it with you. 3 situations can be possible. One is they will not let you to match the TM value that is demanded. Okay, second, they can fall the uniqueness, and third, there can be repetitive structure that can promote self complementarity. Clear? Second important thing, why we are not using low TM value. So low TM value is not recommended because low TM value in turn affect their annealing temperature, and as a result annealing temperature will be lowered and they will promote mismatch priming. Clear? And another important thing, what will happen when we will use too much repetitive sequence in the primer. Okay, so they can promote self complementarity. Not only that, if that sequence becomes complementarity with its primer, then they can promote primer dimer. Both are avoidable. And another type of question that I have seen in previous year questions, that is on the basis of this formula. They will give you a sequence and then they will told you to find out the TM value of that particular primer. Then you have to put the values in that formula in similar way that I have explained you, and you will get your answer. Okay, so that was the video for primer designing. So I hope this video will be helpful for you. Thank you for listening to this class.